Alright, so hello everyone. My name is Ahmed Youssef and I'm a bioinformatics PhD candidate at Boston University. In this video, I'm going to walk you through some of the main parts of my poster titled Deconvolution of Bulk Proteomics using Single-Cell RNA-seq. Now, as the title suggests, this project is interested in deconvoluting proteomics data, and the reason behind that is because in our typical proteomics experiments, we tend to be limited in terms of resolution to the bulk level, losing information on the cellular context within which the observed protein abundances actually occur. And so the goal of this project was to come up with a computational approach that could help us go beyond these bulk level measurements and down to single cell resolution to help improve our understanding of the organization function of the proteome. So now I'm going to start by walking you through some of the main figures here, and if you start with the bottom left, I can explain the experimental setup. And so in our typical experiment here, we start with a number of samples, and these samples could correspond to different time points in a time course experiment, for example, or they could be different tissues or different disease subtypes or so on. And the idea is that each one of these samples is composed of its own unique set of cell populations, each of which has their own unique composition. Now, from these samples, we extract two data sets in parallel. The first one is single-cell RNA sequencing data, which we use to identify the different cell types that are present within each of their samples, as well as quantify the relative proportion within each of such samples. Now, in parallel, we also use mass spectrometry to obtain a proteomics data set, which identifies the total protein levels across each one of these samples. And as such, I now have two different pieces of information here for the same system, which is the cell type composition of each one of our samples, as well as the total protein abundance for each one of the same samples. And as such, we can use those cell type compositions and their changes across samples that scRNA-seq allowed us to obtain in order to uncover what is the relative contribution of each one of these cell types to the observed bulk proteomics measurements. Now, in order to achieve this, we came up with a fairly simple model here, which I'm showing on the bottom left of the screen. And the premise really here is that the bulk proteomics data we observe is essentially the sum of some underlying real single-cell state profiles. And since we already know the number and proportion of each cell type in our samples as obtained directly from scRNA-seq data, we can create a model that looks like this, where we have three simple matrices, two of which we obtained from our previous experiment and one of which is unknown. And by solving for this matrix, we're able to predict those cell-level proteomics profiles. So allow me to move to another part of the poster to elaborate a little bit more about this model. So I'm going to go to the top right here to show a simplified sketch of the algorithm. And as you can see, we are modeling it using three simple matrices. On the left-hand side, you have the bulk matrix Y, which is our proteomics data. On the right-hand side, we first have this matrix we denote as A, which contains the cell type proportions across the different samples as obtained using scRNA. And finally, the single-cell matrix X, which contains the true underlying single-cell protein profiles that we do not have experimental evidence for, but that we seek to predict. Now, given two out of these three matrices, we can use regression to solve for the third unknown matrix. The issue that we tend to run into here is that it tends to be an underdetermined problem. So for example, in a case where we're studying a few subtypes of cancer, there are typically dozens of cell populations involved, and that creates a situation where there are multiple possible solutions. And so our model applies a number of constraints which aim to guide the algorithm to find the most likely combination of single-cell protein profiles that would lead to the observed bulk proteomics data. For more information on the details of the mathematics of the algorithm, I can kindly refer you to the QR code at the bottom here. Now, we have a case study for this algorithm where we studied EMT transition, which is a process by which epithelial cells gradually become mesenchymal, and this is associated with cancer as well as developmental biology, among other interesting areas of biology. Now, in this experiment, we profiled cells undergoing this transition. So they started off as epithelial cells, and over the course of 8 days, they became gradually more and more mesenchymal-like. And for each one of those 8 days, we obtained both proteomics data, for which we have roughly 7,000 proteins quantified, as well as scRNA-seq data, for which we obtained roughly 2,000 cells across the entire time course. Now, to walk you through the pipeline that I just described earlier, we use the scRNA data to identify 10 unique cell states that occur as the cells go through the transition, as well as their dramatic changes in proportion across time. And then using our algorithm, we were able to use those proportions to deconvolute the bulk proteomics data and uncover the contribution of each of these 10 cell states to those bulk proteomics measurement. And as such, we are able to identify the previously unknown proteomics profiles of these scRNA-defined clusters. All of this without relying at all on any level of correlation between the proteomics data and the RNA data. After all, we're only using the RNA data to identify the cell populations that are present, but not to predict the actual protein levels. Those are only obtained from the actual mass spectrometry data. Now, for the rest of the poster, I have some different plots showing off both mathematical validation, biological validation of the results. But I'm going to end my video here. Thank you for listening to my presentation, and I hope we can chat more using the online chat feature.